Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It is entitled Genomic Data Quality, Connecting the Dots Between Bioinformatics and Physical Materials, presented by Dr. Jonathan Jacobs. Dr. Jacobs is the Senior Director of Bioinformatics and BioNexus Principal Scientist at ATCC. In this presentation, Dr. Jacobs will explore open questions on genomics data quality. He will then discuss why ATCC is committed to providing reference quality genomes for items within the microbial collections. He'll then focus on ATCC's approach to whole genome sequencing, assembly, and genome annotation for producing high-quality authenticated reference genomes. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Jonathan Jacobs.
Hello, and welcome to the webinar. Today, we're going to be talking about genomics data quality and the connection to biological materials at ATCC. I'm Jonathan Jacobs, Senior Director of Bioinformatics and BioNexus Principal Scientist, and thank you for joining us. First, a small bit about ATCC. We are a nonprofit research organization which is nearly 100 years old, and we're the world's largest biorepository for bacterial, mycology, viruses, and human and animal cell lines. We also have extensive cGMP biorepository and manufacturing capabilities, and we're a global supplier of authenticated cell lines to researchers in a wide range of industries. On the topic of microbial resources, ATCC has an extensive collection of high-quality molecular-grade diagnostic materials for SARS-CoV-2 and other pathogens, as well as an array of microbiome standards that can plug right into your research program. And we also have a vast collection of multi-drug resistant or single-drug resistant bacterial strains that are available for your research purposes. We also have an extensive range of custom services to provide our customers with both biorepository services as well as production and characterization. And we also serve as an important culture collection for type strain deposits of fungi, bacteria, and viruses from around the world. Lastly, we also have an extensive collection of cell biology resources as well. And I encourage you to take a look at the ATCC catalog if your work is focused on cell biology or biomedical research. And now for the today's talk. We'll be focusing on addressing some current open questions around genomics data quality, particularly as it relates to publicly available reference genomes for bacterial, viral, and fungi. Be providing a few examples of some particularly interesting problems that have occurred with public data, why this matters for your research, what you can do about that, and I'll talk a lot about what ATCC is doing to address issues around data quality and data provenance, and the ATCC Genome Portal. So just as a reminder, Genbank is ostensibly the largest publicly available reference database for whole genome sequencing data in the world. It's also growing at a pace of where it's doubling about every 18 months. This is enormously fast growth. Currently, Genbank has over 230 million sequences for whole genome sequences contained within it. And the raw data that stands behind Genbank, which is NCBI Sequence Read Archive, currently is over about 15 petabytes in size. And although this is very impressive, I think we need to be asking ourselves, how sustainable is this growth for Genbank? And how reliable is the data that's contained within Genbank? And lastly, and importantly, If you have a public resource like Genbank that's growing as quickly as it is, how do you build trust and establish methods or approaches to trust the data in an ecosystem that's this large and growing this quickly? A couple of interesting publications that came out over the last few years highlighted some of the issues associated with this public resource. For example, over a quarter of bacterial isolates, whole genome references for bacteria that are in Genbank that are associated with foodborne diseases are missing key metadata attributes because the depositors just simply didn't bother to deposit this data alongside the reference genomes. And this is a problem for the epidemiology and biosurveillance community because the metadata actually is what is going to help them make inferences on source of origin or the type of media or the context in which the original isolate was collected. About a third, a little more than a third of that sample information is actually lost in the transition from a publication to depositing the data into Genbank. What I mean by that is authors will oftentimes be required by journals to provide very detailed, very enriched data sets along with their primary publications. And when they actually go through the exercise of depositing the data into these public databases, much of that data is also lost. And so you're left with reference genomes that are sort of, in a way, out of context, and it requires a fair amount of work to go back to the original publications and to ferret out and source this information. And lastly, it's a bit shocking here, is there was a study that came out of the Netherlands earlier this year where they surveyed over 8,000 research scientists in the EU. And they actually found that 1 in 12 scientists have admitted to falsifying results within the last 3 years. And so this collectively asks, kind of creates a question around what is the reliability of the data in the public domain, and what is the level of accountability for data that is either missing or omitted, or cases where there's error in the data itself for the depositors, and what is the impact of that long term? I asked myself, when was fake data first deposited into Genbank? And the earliest instance of this that I could find was actually back in 1995. This was a significant event at the time, and it was a case of a graduate student who was very ambitious, had published several papers in top-tier journals, including Science and Nature, and they had deposited gene sequence data into Genbank as part of their research. It turned out, unfortunately, that the data that was produced by the student was false. It was extensively obfuscated and crafted in a way where it would not readily appear to be false, but indeed actually the data was false. And this screenshot here is actually from the United States Federal Register, indicating that this student was no longer eligible to receive any federal funding for a period of 10 years or be employed by the U.S. federal government for a period of 10 years because of this data falsification. And I was curious, though, what happened to this data? Was it actually removed and redacted? It turns out that not only was the data not removed from Genbank, 24 years later, the data and the papers that were actually cited as being false are still being actively cited in the primary literature. And this is an issue because it draws attention to the fact that there's data in Genbank that doesn't just have errors in it, but actually is completely falsified. And it begs the question, what kind of impact has that had on other people's research? 42 citations, in fact, still cite this data. And the data, here's a screenshot of the sequencing data that was provided by this graduate student for the original sequence for the human neurofibromin gene, NF1, and its promoter sequence. And this is not just something that happened, 25 years ago. Actually, there was a case just a few years ago where not only did multiple instances of falsified mitochondrial DNA for a range of different bird species get deposited into Genbank, but this was actually part of been supposed by this publication that I'm showing here, that this falsified data was deposited into Genbank for the purposes of constructing a falsified phylogenetic tree, because it was essential for this lab to draw relationships between otherwise unrelated bird species for their funding, for continuation of their grant funding. So they crafted an extensive series of false mitochondrial genomic DNA, deposited that data into Genbank over a period of a few years, and then eventually they were able to draw phylogenetic trees that connected these species together when in fact data does not support this and the data itself is actually falsified, is fabricated. But that actually is still in Genbank. And it is labeled as being unverified. But it's important to know that under very rare circumstances is data ever removed from Genbank. The original depositor is the one that has to initiate this data removal and redaction. And instead, what can happen is that data is labeled as quote-unquote unverified. But nonetheless, the data is still there. And it begs the question, what kind of impact is that having on others who are doing research in this particular space, who maybe discover this sequence through a blast search or some other bioinformatics study, and unwittingly unaware of the fact that this data is false or quote-unquote unverified, what kind of impact is that having on their work? So there are also other issues beyond the higher profile cases of intentional data falsification, of course. We all know that biology is messy. Accidents happen. But several studies over the last few years, and this is just 3 I'm showing here of over half a dozen actually, that looked at the quality of the data in RefSeq and in Genbank and identified a variety of different types of large-scale contamination. In one case, the researchers on the publication on the left here identified that a significant number of bacterial genome references in RefSeq are actually contaminated by the PHI-X genomic sequence. And this is because Illumina sequencing, when you do a library prep, you use PHI-X as a loading control, and the sequence from those PHI-X genomes actually have made their way and percolated through the bioinformatics pipelines and actually are sprinkled throughout a large number of bacterial genomes, which of course are negatively impacting the quality and reliability of those genome references. Another group identified that there was actually a large amount of human genomic DNA contaminating bacterial genome references. And this is in turn spurred the creation of thousands of spurious protein sequences, because genome references themselves are used as a backbone for predictive analytics to create databases of predicted protein sequences. And if you have bacterial genomes with human DNA in it, DNA sequence in it, then those proteins themselves are being labeled as originating from a bacteria, but when in reality they're either not real proteins at all, they're chimeric proteins, or they're actual proteins that they're only found in mammalian or in the human genome. So either way, it's a problem because the taxonomic identity of those proteins is incorrect. And lastly, there's been a substantial number, over 2 million actually, cases of the 230 million or so genomes in Genbank are actually contaminated with sequences from other species other than PHI-X or human. And it may not sound like a lot out of the whole 230 million or so, but 2 million genomes contaminated with sequence data from other organisms is still a lot in absolute numbers. And whether or not it impacts your research or not is an open question. Obviously, it depends on what genomes what organisms you're working with. Have those genomes been removed from Genbank? No. Are those genome references labeled as having contaminated entries in them? No. How would you know to find out if a particular reference genome that you're working with is contaminated with sequence data from another organism? You'd have to do the bioinformatics analysis yourself, or happen to cross paths with one of these publications and take a close look at the metadata associated with these publications, or use the bioinformatics tools that were developed as part of these research programs. Either way, the key thing here is, these types of publications really beg the question, how do you create high quality reference databases that are actually dependable, not contaminated, and don't have these chimeric chromosomes or genome assemblies present in them. How do you depend and trust the genomics data that you're obtaining from the public domain when so many of them have been identified as having systematic problems like this that remain and persist in these databases? And lastly, I'll mention that there have been cases where entire genomes are misclassified and assigned to the incorrect taxonomic clade. Roughly about 4% of the entries in Genbank are in the wrong genus, and close to 8% of them are misclassified at the species level. And this may seem like kind of minutia to some, but for those that are studying comparative genomics and microbiome profiling, or are actively engaged in microbial genomics research, this can have huge impacts on the conclusions that you make from your own analysis. And so again, actually, when you think about close to 8% of 240 million reference genomes in Genbank, that's 8% may not seem like a lot, but that's hundreds of thousands of reference genomes that are actually in the wrong taxonomic assignment. So it's a bit of a catch-22. So many of you listening to this webinar sort of have an idea of what the overall workflow is, but just to cover it here briefly, you start out with source material, your biological source material, go through DNA extraction, library prep, sequencing. The raw sequencing reads go into a Bioinformatics Pipeline for a genome assembly, novo assembly, and annotation. And then the data is submitted to Genbank as a new draft assembly for species X. Interestingly, the way in which Genbank actually assigns taxonomic identity, the criteria it uses for doing a contamination check, and a QC on these de novo assemblies actually is based on data that's in RefSeq, which is a subset of Genbank. So if you submit a reference genome that's very, very high quality and it makes it into Genbank, it may potentially also make its way into RefSeq, which then in turn will be used to help vet and sort of filter and screen incoming genomes in the future that are related or near neighbors to your genome reference that you've sequenced. The problem is, though, is that when data makes its way into RefSeq, when genome assemblies make their way into RefSeq that are not actually high quality, for whatever reason they may have passed the bioinformatics checks, but the taxonomic assignment was incorrect, or some of the metadata associated with that genome was incorrect. This can actually negatively affect the future data that gets submitted to Genbank, because there's a cyclical relationship here. And NCBI is doing a fantastic job trying to develop automated methods and systems to not just annotate these genomes, but to also do a series of QC checks on these genome assemblies, but they simply don't have the staff to manually view and vet every single reference genome that comes into Genbank. So they rely substantially on these automated methods. In fact, the majority of the metadata associated with these genome assemblies actually is, there's no requirement for the depositors to even provide a lot of the metadata that most researchers would kind of assume is required, like where did the material come from? How is it isolated? What was the conditions under which it was grown and maintained? Was it part of an experiment, a mutagenesis experiment, or is it an original isolate that was obtained from an environmental source and it's otherwise untouched and has not undergone any lab adaptation? Was it obtained from a culture collection directly, or was it passed from one lab to the next for being deposited and sequenced and deposited into Genbank? All of these questions are sometimes answerable based on the metadata that's associated with the individual assemblies, but oftentimes it requires a lot of manual work to kind of uncover these details. And actually, one of the things that we uncovered in our own analysis is that there is a correlative association with the amount of metadata that's associated with an individual genome assembly, and the completeness of that metadata correlates very nicely with the quality of the genome assemblies themselves. What this means is sort of self-evident, but it's, researchers who care about details. They produce high-quality work, and those genome references tend to go into RefSeq. However, there are still many examples of incomplete or chimeric or taxonomically incorrectly assigned genome references that are still maintained within RefSeq. And unless you know what you're looking for or have the tools available to you to actually discover and vet these samples, these assemblies, it can become very difficult to know whether or not a particular genome assembly or collection of assemblies out of RefSeq is actually going to negatively impact your work or not, or whether they can be trusted and relied on. So, you have to tread with caution and sort of trust but verify. So there's a number of factors that contribute to genomic data quality issues. Misclassification, chimeric genome assemblies, sample contamination, sequencing errors from the instruments itself, of course. The scientists that deposit the data sometimes mislabel data, making honest sort of data errors. Sometimes data is omitted on purpose for a variety of reasons. Sometimes the data is obfuscated in a way that makes it difficult to ascertain the origins of the data. And then, of course, there are these sort of higher profile cases where there's been intentional misconduct and falsification of the data. And these issues affect the entire spectrum of the life sciences and research community, because genomics and bioinformatics is increasingly becoming a foundational discipline that affects all of the research going on in life sciences around the world. And these factors need to be addressed, and there's a tremendous amount of work to be done. And I'll talk next about what we're doing at ATCC to help address some of these problems. So before I get into that, just as I'm discussing what we're doing at ATCC, keep it in your mind these questions, right? What's the cost of poor quality data that's in public genome databases? And what are the consequences for sharing, quote unquote, bad data? How is accountability for contributing imperfect data held up by the research community? And what are some things that you can do to actually help address these issues? So I've come up with sort of 4 ways individual labs can help improve the quality of the genomics community's data. First, my recommendation is that everyone should be sort of taking the mindset of trust but verify, right? What I mean by this is that the majority of data that's out there in the public domain is of high quality and is reliable and is fine to use. But wherever possible, you should be using authenticated source materials, not lab-adapted strains or materials obtained from another lab. The data itself also should be authenticated. Don't just assume that the data, genome reference, for example, comes from the original culture collection if it bears a culture collection identifier. Oftentimes those reference genomes with culture collection identifiers as a strain name are handed down from one lab to the next. I'll talk a little bit more about that in a minute. But lab adaptation can have a real serious impact, not just on the variability of the resulting reference genomes, but also on the phenotype of organisms. Second, be a standards champion. What I mean by that is be aware of what the standards are that affect your research, and what are the standards that are out there associated with microbial genomics, and adhere to those standards, uphold those standards. If you're submitting reference genome to Genbank yourself, use the MIGS specification or one of its variants to establish what kind of metadata you are going to be depositing alongside the actual genome assembly itself. And if there aren't good standards for the research area that you're in, get involved with the Genomic Standards Consortium and help define new ones, because standards are one way that we're all going to be able to benefit from improved quality in the metadata associated with these public genome reference databases. As it's also important, number 3, to assume that any data coming from outside your lab is dirty. Data itself is rarely clean. Public data is probably even less clean. So it's important to recognize that and to take the appropriate steps to clean and vet data before you incorporate it into your own research program. A student once asked me if it was okay just to download RefSeq and use it as is. And I said, mostly, just sort of depends. And the problem is you don't know if publicly available data is going to be a problem oftentimes until after you discover the problem. But if you vet the data up front, you can mitigate a number of those issues ahead of time and make sure that you're working with data that's clean and fit to purpose for what your research program is. And so that ties into this last part about having a digital mindset. What I mean by that is throughout my career, I've, as a bioinformatician, often brought in a research program at the end. After the data has been produced, after the experiments are done and the pipettes have been put down, they then turn to the bioinformatician and ask them to analyze the data. And if the data itself is of poor quality or the experimental design was not appropriate, sometimes you're kind of in a bind as a bioinformatician because you're dealing with data that's of poor quality or experiments that didn't really work out and they didn't realize it until after the analysis was done. So involve the bioinformaticians early in your research programs. Ask them and reach out to them to vet and clean the data, verify its authenticity and the source, and build standardized sets of pipelines ahead of time so that you can identify pitfalls or other issues that might crop up during the course of your research program. All of these things will sort of tie together and improve the reproducibility of your research and also for you and the rest of your team kind of help everyone have a better eye for quality and to identify issues where quality may not be what you're expecting. So on the topic of trust but verify, I just want to walk you through a brief example. So I've changed the names of some of the folks involved in this story just to sort of protect the innocent, if you will. And I've replaced them with names of folks that are actually in my own lab. So in the early 70s, ATCC received important type deposit from a prominent researcher in the microbiology research community. And this, we'll say it's the Benton lab. This lab, then the PI of this lab who deposited these materials with ATCC, in parallel, also handed those samples off to a postdoc that went on to start their own lab, who then handed those same strains off to additional postdocs and other graduate students in their lab over time, who went on to have their own labs down the road over a period of 30 years. And eventually, in the early 2000s, one of these researchers realized that this particular strain had never been sequenced before, pulled it out of their freezers, did whole genome sequencing in 2009 and deposited that reference genome into Genbank. This initial genome assembly became a kind of the giraffe reference for that species. Meanwhile, in the mid-90s, there was a request that came in from a separate lab to ATCC to obtain this strain. Totally parallel lines. This researcher obtained the strain in 1995 and again conducted a series of research on this over a period of 20 years. And they sequenced it themselves, and what they noticed is that their version of their reference genome was actually different from what was in Genbank. So they went ahead and deposited their version of the genome reference into Genbank using newer technology. And because of the differences in the technology platforms used, Genbank largely admitted this sequence in to replace the previous one, and it became a new high-quality closed reference genome. The problem is that these two labs both had what they thought were the same strain, but when they looked at the phenotypic data, these two strains were behaving very differently in the lab, separate from the genomics data. Not to mention that there actually were bona fide differences in the genomes. So they reached out to ATCC and they said, hey, we need to sort this out. So we went ahead and sequenced these materials ourselves using both Illumina and Oxford Nanopore sequencing. We did a hybrid assembly, and we sequenced to completion the original source material that was actually deposited into our collection. And what we found actually were over 10,000 SNPs in this relatively small genome between our in-house reference and the high-quality reference genomes in Genbank. And over 13,000 SNPs differed from the original draft that was deposited in 2009. And the issue here is that these strains had an unknown chain of custody. They had potentially undergone a significant lab adaptation in both of these labs. And the end result was not only were the reference genomes different, but the two strains were actually behaving differently in the lab. They had different phenotypes. And you know, that's a problem for researchers out there that are looking to have reproducible research. So in 2009, on the topic of being a champion of standards, there was a significant effort to establish what's called the Minimum Information for Genome Sequence Specification, or MIG specification. This is one of the data packages that has governed how metadata is provided by Genbank whenever someone deposits a new reference genome into that database. But what was interesting about this initiative was that the requirement for having source material as an identifier associated with every reference genome that adheres to the MIG specification was actually not required, even though the group recommended that this be a core descriptor. It is actually not a requirement for someone who's depositing a reference genome into Genbank to actually provide data or information about the provenance of the physical biological materials that were used to produce that reference genome. In fact, in the original paper, they even say, quote unquote, physical archives are still not yet routinely created by researchers. And that highlights the important problem here, is that even if there is a reference genome in Genbank, if you can't go back to the original biological material to resequence it, to actually do research with that organism in your lab, it begs the question of what is the authenticity of those reference genomes to begin with, if you can't trace it back to physical materials. Assuming the data is dirty, well, this is another point I'd made earlier. So at ATCC, as part of our comparative genomics study, we've found that there are a large number of bacterial strains in Genbank that bear ATCC strain identifier names. On the left here, for example, you can see Acinetobacter baumannii 17978. There are actually 7 different assemblies in Genbank for this strain. And what I want to draw your attention to here is the bottom row within that red box. And you'll notice that between of these 7 strains, one of them, the last one in this set, has over 60,000 differences, SNPs, compared to the reference genome that we produced, and over 2,400 insertions or deletions in the genome. And this, again, should highlight the fact that these genomes, these 7 different assemblies, are all different. And I would argue that this number 7 on this list is perhaps not even the same strain, let alone, it clearly had a different natural history from the other 6. What we're doing at ATCC is we've also adopted this digital-first mindset. We have a completely controlled end-to-end workflow where we're, from the point at which we obtain materials out of the ATCC biorepository, all the way through to the point in which we do whole genome sequencing and releasing of those genome references at the ATCC Genome Portal. We're tracking every step of the way all of the metadata associated with the samples, and they're using a quality-controlled process for isolation, for genomic DNA extraction, our sequencing, and our bioinformatics analysis. So we have a standardized set of workflows. All of the data is being tracked, and for anyone that's interested, they can trace the genome assemblies that we're producing all the way back to the physical materials themselves. So the elephant in the room here, in my opinion, is that, is around data quality and provenance. I believe that these are some of the 2 biggest issues facing the genomics research community for the next 5 to 10 years, and there's a significant amount of work that organizations need to do to address these problems because it's eventually going to come around and have significant costs for research programs that end up at dead ends or come up with false conclusions of their research because the data that they're using from the public domain is of poor quality. And we're looking to address that at ATCC. And now I'll talk a bit about the ACCC Genome Portal. So we question how many quote-unquote ATCC strains are actually contained within Genbank or within the RefSeq, the high-quality reference database that's a subset of Genbank. And we found that close to 2,000, actually this number is a bit higher, this slide is a tad outdated by a few months, but there's actually over 2,000 now strains contained within RefSeq that are labeled as being ATCC reference genomes. It's important for the research community to know that ATCC does not deposit reference genome assemblies into Genbank, and that all of the reference genomes contained within Genbank that bear the ATCC strain name were deposited there by researchers around the world that are using our materials in their research and have sequenced it themselves using their own protocols and methods in their own labs, their own bioinformatics methods, their own bioinformatics quality control and acceptance criteria, and they're depositing that data into Genbank and identifying these strains as being a quote-unquote ATCC strain. But the other part of this to keep in mind is that for those 2,000 references that are in Genbank, in RefSeq, that bear the ATCC identifier, the traceability of those strains actually is pretty poor. A large number of those organizations that have deposited ATCC strains into Genbank, actually, we have no records of them ever purchasing or obtaining strains from us. So how those strains made it into the hands of the researchers that deposited that data into Genbank is unknown. And what then is the natural history of those strains? How many times have those strains passed from one lab to the next before they were finally sequenced and the data was deposited? In 2017, we began planning around this idea of building an ATCC reference database of our own materials and providing the research community with genome assemblies for our bacterial collection. We expanded that to include our microbial collection, both fungi, viruses, and bacteria. And in 2018, the board of directors committed to moving forward with this initiative, where we're essentially seeking out to create what we call an enhanced authentication initiative to verify our materials through whole genome sequencing and to provide the research community with the theoretically optimally best quality genome assemblies available for these organisms. And that the work has been continuing since 2019. You can visit the ATCC Genome Portal today at genomes.atcc.org and browse through our growing collection of bacterial, viral, and fungal genome references and download those reference genomes for your own research use, including the annotations, because we do whole genome annotation as well. And there's a lot of additional development work that's going to be going on in the future to expand both the content and the tools that are going to be included in the Genome Portal. So you can not only just search for organisms based on taxonomic names, but there's also some BLAST-like tools there where you can actually submit your own sequence data to the Genome Portal and use that sequence data to search for and find if there are other organisms that we've already sequenced. The original materials that we sequenced from our collection, also the metadata associated with that is available, as well as all of the metadata associated with the type of sequencing technology we used, the quality metrics that were obtained, etc. And again, just as a reminder, we have a complete end-to-end workflow to maintain data provenance from the point in which a physical sample is taken out of our repository through to the point in which we release the reference genome assembly to the ATCC Genome Portal. So in the context of producing high-quality reference genomes, I'm going to dig a little bit deeper into each of these individual steps. So for the extraction of genomic DNA, we have a limited number of protocols that we use that have been validated. Our goal is to obtain high molecular weight, long, large fragment size genomic DNA from these materials. And for our bacterial extractions, the vast majority of the extractions that we do involve using the QIAGEN genomic tip technology, for example, because that produces very long DNA fragments, over 50 kb very often, which is ideal for sequencing on the Oxford Nanopore long-read sequencing platform, which is part of our sequencing, which I'll talk a bit about in a moment. We also have very hard thresholds in terms of acceptance criteria for purity and fragment size, and any sample that comes through our extraction lab that doesn't meet these criteria is discarded. This is an example of our fragment analysis that we do on every single one of our samples. All of this data is available on request for every single genome reference that we have. And you can see, for example, in this particular example of Corynebacterium, our average fragment size for this library prep was over 42 kb. We collect all of this data for all of our strains, including DNA concentration, purity, fragment size, and it's available request for our entire collection. In terms of the sequencing technology, we sequence all of our bacteria and fungi on both Illumina and the Oxford Nanopore Technologies platforms. This gives us the best of both worlds. By focusing on the Illumina technology, we're getting short read, high quality sequencing data, which is really important when you're sequencing near neighbors of different strains, and the individual base calls and at the SNP level resolution are key. The long read technology from ONT provides us with the ability to produce closed, for 98% of our samples, very, very high quality genome assemblies that are superior to what's found in RefSeq. And so we use both sequencing technologies, combine the data together into a hybrid assembly pipeline, which I'll talk more about in a moment. And this is an example of some of our sequencing QC results. The top row here for 5 of our different isolates, you can see the Fred scores for our Illumina data, and our cutoffs for acceptance criteria is anything above a Q30. And on the bottom row for the same isolates, you can see our Oxford Nanopore quality scores. We have a Q10 cutoff there for ONT data, and this is kind of an overview of the read length distribution versus the quality of the reads themselves. So the advantages of a hybrid genome assembly approach, where we're using both of these technologies, is sort of illustrated here in this diagram. This is called a bandage plot. And what you're looking at on the far left is a genome assembly using only the Illumina data. And what this is each of those green segments are contigs with unknown connections to other contigs within the same genome. Now, we all know from a biological standpoint that this particular genome is a circular bacterial genome. It's supposed to be one continuous stretch of DNA that forms a loop, right? Forms a complete circle. But there's a lot of uncertainties in these genomes when you do a de novo assembly, because there's long tandem repeat regions of genomes. There's certain regions of genomes that are much more difficult to sequence than others. And so scaffolding each of these individual smaller contigs together into the proper order that would actually create a circular loop is difficult to do when you're only relying on Illumina data. And so the Oxford Nanopore data is included in this assembly, and what this does is it allows us to create ever increasingly more complete circularized closed genome for all of our bacterial references. And this is another couple of examples here of 4 different ATCC strains. On the left, for each group, you can see what the genome assembly would look like with only Illumina data. And on the right, you can see that with the addition of the Oxford Nanopore data, we're able to close these genomes completely and provide one complete reference, finished and closed reference. So now I'll talk a bit about the Bioinformatics Pipeline. We work closely with our partner, OneCodex, a cloud-based metagenomics profiling bioinformatics service, and we're using their computational backend to where both ONT and Illumina data is uploaded to OneCodex. The raw reads are profiled using a Camer-based taxonomic profiling tool, which also removes and eliminates potentially contaminating reads from other organisms, which doesn't happen very often, and it's usually well below 1%. But nonetheless, it's important because any kind of contaminating reads that would make its way into your actual de novo assembly could bias the assembly or produce chimeric contigs. We then go through hybrid assembly for bacteria and fungi. On the viruses, we only sequence on the Illumina platform. And that's because the vast majority of viral genomes are smaller, and it's very often that we're able to create closed, finished genomes using only Illumina data for our viral genome assemblies. So it's not necessary in many cases. There's a series of different quality assessments that are done after post-assembly, looking at the total coverage, our expected completeness based on near neighbors that are in one codex's database, and an assessment of contamination, if there is any. If those assemblies pass our QA assessment, then we go into a genome annotation phase, and then we make a determination on what the quality level and assembly level is for the final assembly. And on the bottom here, you can see these are some of the specific bioinformatics tools that are part of this pipeline along the way. This number is already out of date. As of today, we have over 1,600 ATCC Standard Reference genomes available through the ATCC Genome Portal. Last year, in 2020, we did over 1,700 extractions and over 1,600 sequencing libraries, 331 sequencing runs, and we finished over 1,000 genomes in 2020. And we're on track for 2021 to exceed over 2,000 by the end of the year. Our current collection contains about 2/3 of them are completely circularized, closed, finished genomes. About a quarter of these genomes are important, particularly for folks in the public health space because they represent type strains. And 147 of these are multi-drug resistant strains of bacteria, and 56 of them are considered microbiome standard reference genomes that are used in the research community, both in bioinformatics context and the physical microbiome standards themselves or products that are sold by ATCC that are available for these as well. And we're updating this number every month, and we typically release around 100 or a little over 100 genomes on a monthly basis. This data here, kind of on the left, illustrates some of the depth and of the number of sequencing reads produced on Illumina or for ONT or Oxford Nanopore for the same samples. Each one of the dots on the plot on the left represents a different genome assembly. And the reason there are like two clouds here is because we also use an Illumina NextSeq as well as a MySeq. The NextSeq is much higher throughput, tends to generate a lot more data for individual samples, and we're working towards an automation approach for our library prep where we'll be able to get even more samples on our NextSeq runs because we have the bandwidth to do that. On the right, you can see on the y-axis the percent completeness of those genomes. This is a CheckM measurement, and you can notice that we don't accept any genomes that are below 95% complete. And the x-axis is a ratio of the N50 value, which is sort of a measurement of the degree of fragmentation of that genome assembly over the genome size. So as N50 over genome size approaches 1, you're getting closer and closer to having a complete genome. Now, not all of our genomes are completely closed and complete, but over 95% of them are. And as far as the overlap of this data with what you find in RefSeq, interestingly, about 1/3 of the ATCC Standard Reference genome assemblies represent strains that are already in the public domain, that are in NCBI's RefSeq database, 366 at the time that this data was collected. Strains are actually represented in both databases. So importantly, we've contributed 747 new genome assemblies for bacterial strains that have never been sequenced before. And we have plans to look closely at the strains that are in Genbank that have genome assemblies for them that are of poor quality, and we're going to systematically go in and sort of prioritize those to resequence them out of our own collection as well in the near future. The plot on the left here, the scatter plot on the left, shows a comparison of the N50 values, or the level of fragmentation of these assemblies in our data versus data obtained from RefSeq. And the vast majority of these points are actually along the diagonal, but the important takeaway of the figure on the left is that 98% of our sample, over 98% of our assemblies actually are more complete and are higher quality than genome assemblies that are in RefSeq for the same strains. So in summary, public genomics data should be handled with care. It's usually okay. It often has errors or omissions The quality of the genome assemblies are oftentimes not the best. And when you're dealing with public genomics data in aggregate as part of a large database, the issues around data quality, potential data falsification, and the metadata associated with those individual entries in that database could really potentially impact your research. And so it's important to remember to trust but verify the data. And if there are particular instances in a public database of reference genomes that really matter for your research, be curious. Do the additional level of investigation to verify the quality of those reference genomes. Where do they come from? How are they assembled? Is the genome assembly actually up to snuff? Is it at a quality level that you would accept if you were doing the sequencing in your own group? The ATCC Genome Portal, we're currently the only genomics database with 100% data provenance for every entry in our database back to a physical biological material that is still maintained in a repository, and that's available for, it's available from ATCC. Over 98% of our assemblies are superior to those that are found in RefSeq, and we're adding over 100 new genomes every month, and we're always looking for suggestions. So if you are interested in having us sequence something in our collection, please contact us and let us know, because we would be happy to put those requested reference genomes at the top of our list, top of our queue, and get those genomes out there if it matters for your research. And with that, thank you very much. Please feel free to contact us if you have any questions, either by email or via social media on Twitter. And also, if you're interested, we're also hiring. We have ATCC has a number of positions that are open, both on my team directly and as well as in our biomanufacturing and our research and development divisions. So please reach out, and remember, stay curious. Thank you.
All right. Well, thank you, Jonathan, for the excellent presentation. In just a few moments, we will begin our Q&A session. As a reminder, please use the chat function in the webinar program to submit your questions. And it looks like we already have quite a few questions come in, so let's go ahead and start with the first one. So Jonathan, with the information that the Genome Portal provides, are blast searches in the NCBI database to be trusted?
I think in general, most of the data that's in Genbank or NCBI is trustworthy. I mean, people there's not really very many instances of intentionally messed up data in Genbank. The challenge is that, and we're finding this increasingly to be the case, is that you don't know ahead of time the source or the sort of history of search results that you would get back from a blast search. Like you get a hit on a certain gene or a certain genome, who produced that genome, under what conditions, how reliable is that genome reference. We recently published a paper in ASM's M-Sphere journal where we actually looked at about 1,000 genomes that we had, bacterial genomes that we had sequenced and assembled on our own that are available through the Genome Portal. Actually, there's more like about 1,700 or so bacterial genomes there now. And we compared that data to the same quote-unquote strains, the same labeled strains for those same genomes in Genbank. And we found quite a few issues with not just differences in SNPs or individual mutations, but in some cases there were kind of gross structural genome changes and differences. In one extreme example, for example, there's a strain of Acinetobacter baumannii in Genbank that is about 60 or 70,000 SNPs different from the rest of the entire clade. And it's probably actually a misassigned genome. It's very likely a different species. So I guess at the end of the day, in general, usually, most of the time, the blast results you get back from a NCBI blast search are reliable. But if you get a hit on something that is also represented in the Genome Portal, then it would be good to kind of vet those results to see if the sequence is actually the same sequence that we have. And one thing I'll add here is that we have it on a roadmap to actually probably won't be until later this year in Q4, where we're going to be rolling out an integrated, essentially taking RefSeq from NCBI and then doing an inline replacement of the genome references that are there, where we'll be integrating in the genome sequences that we're producing and then re-indexing them so people can run those blast searches on the Genome Portal and make sure they're getting accurate results.
Okay, good. So kind of as you mentioned in the talk, trust but verify, right?
Yeah, exactly. Yeah.
Okay. You mentioned we had a certain number of genome sequences in the webinar, but this was pre-recorded, right? So how many genome sequences are we at this moment?
As of today, we are around 20, actually I'll just pull it up. We have about 2,163 genome assemblies in the Genome Portal right now, and of those, about 1,840 are bacterial genomes. The rest are fungal and viral genome references. And we're going to be we're adding about 100. We like to add about 100 a month. Some months we do more, some months a little less, depending on what else is going on. But what's interesting is that we're also getting into sequencing microbial eukaryotes like protists, trypanosomes, and others. Some amoeba, we sequenced an amoeba recently. So we're going to start to see, end users of the Genome Portal are going to start to see a wider variety of organisms as well.
So that's very exciting, actually. There's kind of a follow-up question to this. Are these fully finished genomes with one or fewer contigs/assemblies, or are they drafts of multiple contigs?
Well, some of them are fully finished and complete. It really just sort of depends. We kind of have our own rating system where, and acceptance criteria on what we consider to publish. Generally, we shoot for a minimum of 100x coverage for our Illumina data and 40x coverage for our Oxford nanopore data that we're producing for each bacterial genome and fungal genome. That, for most cases, results in a quote-unquote complete genome. We also use a tool called CheckM on the bacterial assemblies, so we don't accept anything that's below 96% complete based on CheckM rating. So it's possible that there might be little pieces that are missing, but by and large, the genomes that we're producing are either complete or would be considered a high-quality draft. When we did our comparative genomics analysis last summer, again, the results are which are in that MSphere paper. There were very few genomes that were in Genbank, which had a superior assembly quality compared to the ones that we ourselves. And that's really kind of what we're shooting for, right? We're trying to make sure that the data that we're providing the community is sort of gold standard for what's in our physical collection.
Okay. Now, you may have kind of covered some of this in the last question, but can you comment on what is your threshold for genetic variability between lots of the same product, or even between the master stock and derivative product?
Yeah,
so I guess, in other words, yeah, if you recall the genome standards, what level of finishing is done on these?
Yeah, so all the sequencing that we do is we don't release anything that it would be below quote-unquote high-quality draft. As far as the variability from lot to lot, currently right now, what we're trying to do is sort of go broad, but not deep. So we're not actually doing sequencing on every production lot that comes out of our bioproduction facility for the same strains. Now, it's a big focus of my group right now is on streamlining our processes and transitioning to a fully automated workflow from extraction through library prep all the way into our informatics pipelines. And what that's going to enable us to do is to cost effectively actually do whole genome sequencing on every single production lot. So my expectation is that in the future, we're going to have these reference genomes produced for each time a new strain of Bacillus subtilis goes into production from the same seed lot or same master cell bank. We would resequence it and have that and start to collect that kind of data. What is the level of variability? And for anybody that's been in the microbiology space for any amount of time, they know that for some strains the genomes are relatively stable. Others undergo lab adaptation and are sort of highly variable. So we're basically building the foundation for us to be able to do that kind of long-term comparative analysis now. And so in the future, we're going to start to have that kind of data, which will be really interesting.
Okay, that's very impressive. So in the next question, it goes, I note that your annotation for bacteria is done using PROCA.
Yeah.
Is this a once-off annotation, or for organisms with an active research community, are the annotations updated?
Yeah, that's another. So we don't we update our reference database for PROCA on a quarterly basis right now. It is on the development roadmap for the bioinformatics team to actually implement an improvement. Essentially, you could think of it as like PROCA Plus, if you will, where we're going to be engaging directly with research communities that are focused on certain groups of organisms and actually integrating that input that we're going to collect from our end users into the annotation pipelines for all the species that are in a particular genus, for example, because there are a lot of every clade is a little different. So my recommendation to end users is that the sequence is, I'll stand by that 100% all day long. But the annotations that we have right now are generally speaking good enough for most research applications. However, if you're a researcher that's focused on a particular clade and you have a need for higher quality or deeper annotation of those reference genomes, then you can download the sequence, download our assemblies, and of course you're welcome to re-annotate it on your own as needed. Or if you don't have the capability to do that, or if you have a question about a particular gene, feel free to reach out to us. We're actually right now hunting down annotations for some genes in Acinetobacter baumannii, working with a group at George Mason University on that. So these are things that we do on a regular basis to help the end users actually make better use of the data we're producing.
All right. Now, for comparison purposes, does the assembly and annotation pipeline use a standard linearization and orientation of circular genomes?
Can you restate the question, Brian? I'm sorry.
Sure.
You dropped out there for a second.
Oh, okay. For comparison purposes, does the assembly and annotation pipeline use a standard linearization and orientation of circular genomes?
Yeah. Typically what we'll do is our reference, once the genome assemblies are created, we work with our partner One Codex and use a pipeline that they have to make taxonomic assignments and basically identify the nearest neighbor within Genbank. And we use that reference to basically scaffold or organize the contigs in the same linear order so that there should be comparisons across, but obviously if you have a scaffold that's the sequencing data indicates that there's actually a rearrangement, and this actually happens fairly often. that the nearest neighbor, the reference that's in Genbank, has either it was much more fragmented because it was only sequenced on Illumina, so it doesn't really have good scaffolding, or we actually find that because maybe they were using, at the time they sequenced it, older sequencing technology, the scaffolds are out of order, the synteny, if you will, is out of order. So in cases like that, we don't break contigs in order to put them in and realign them with what the nearest neighbors. We use our own assembly and we don't break those. But as best we can, we'll order those contigs in the same way that the nearest neighbors' contigs are arranged as well.
Okay. We have one final question that has come in. If you have any questions, though, feel free to submit them as we're going through this, but we'll call this last call. So, Jonathan, when you are evaluating contamination of a bacterial specimen, how do you separate potential contamination versus cross-species recombination? And what reference sequences do you use for annotations?
Yeah. So for the first question, all of our raw data actually gets passed through the One Codex Metagenomics pipeline, and we bin our reads so that we are basically we're filtering out any reads that are identified as coming from outside that species. Now, there are what's interesting about the project is that there's actually a number of, especially on the fungal side, there's a number of organisms that we've sequenced that have never been sequenced before. So in those cases, we actually elevated up to the genus level. And I think in one case, we had to go all the way up to the family level in terms of acceptance of the reads. And that's essentially how we identify, quote unquote, contamination. If there is, I don't remember off the top of my head what the cutoff is, but we do have a hard cutoff for the percentage of contaminating reads that we might potentially see in an individual library, which is, I believe it's around 1% or less. It's fairly stringent, which is another thing I think that adds to the quality of the assemblies that we're doing, is that we're being fairly rigorous in terms of excluding reads that really shouldn't be there, because especially if they're from a near neighbor, they can really mess up an assembly as a result. And as far as how do we separate potential contamination versus cross-species recombination, that's an interesting question. Right now we're not really drawing that distinction, but I would encourage the person that asked the question. I think I'm looking at the web interface now. It looks like Kim Gurnett asked the question. So feel free to reach out to me, and I'd love to have a conversation about what you mean by that, because we're always looking for ways to improve the way that we're producing these assemblies. As far as what reference sequences we use for our annotations, I mean, right now we're using the current, the latest, I guess, version of the PROCA annotation database. Going back to my comment earlier that we've been really focusing most of our development efforts on the upfront genome assembly problem and making sure that our assemblies are as accurate as possible. And there's a couple of members of my team now that are transitioning into looking at the downstream annotation issues to see where we can improve things there. And once we make and roll out our improved annotation pipelines on the bacterial genome assemblies we have, the plan is actually to re-annotate everything that we've annotated in the past, which will be another significant difference from the public databases like Genbank, is that typically genome assemblies go into Genbank, they don't ever get re-annotated unless the depositor, I believe, requests it to be done. So we're going to be building a system where essentially, as we improve our own pipelines, we're going to be reanalyzing and improving the quality of the assemblies that we have and the annotations that we have.
All right. Good. Now, two more questions have come in, so definitely earning our keep today. So for this next question, does ATCC and NIST work with each other to provide reference genomes from bacterial and fungal strains? And then could you further comment on how the two organizations differ in their approach to provide the high-quality reference genomes?
Yeah. So we work pretty close with NIST on a number of standards initiatives, like the International Multi-Omics Microbiome Standards Alliance. INSA is an organization that's led by a couple of members from NIST, and we participate in that, and we both have a common interest in making sure that the highest quality reference genomes are made available to the research community. As far as specific differences in our approaches, both in the lab and from an informatics perspective, I can't really speak to that because I'm not entirely sure off the top of my head how NIST does their sample preparation or library preps, and also how their bioinformatics pipelines are built out. I'm assuming this question is related to the NIST standard reference genomes and reference materials that they have produced for certain bacterial organisms. But yeah, I mean, I probably should look into that and get a better idea of how they're doing it, but I can't really speak to it right now.
Okay, fair enough, fair enough. So last question. This may be available on the website, but is there API access to the ATCC reference genome data?
Yeah, that's a great question. It's currently there is, where it's in active development. We have an API that you can get some information about on our GitHub site, ATCC Bioinformatics. Look for that repository or that organization on GitHub. There's a repo for the API. We haven't actually been we haven't added details about the API directly to the website yet, because we're kind of thinking of it as sort of an early access utility right now. But I understand that it's going to be of high interest and high importance, especially to the bioinformaticians out there that don't want to have to go through the hassle of point and click and downloading individual reference genomes. That if they want to grab everything, or more importantly, potentially, is the metadata that we're going to be adding into the Genome Portal in the future also will be all queryable and accessible through our API. So if the person who asked the question has any issues on finding details about the API, again, feel free to just shoot me an email and be happy to get you set up and help you get access to the data as well.
All right. Well, with that, I'd like to thank you, Jonathan, for the excellent presentation and fielding all these questions. And thank you, everyone, for attending the webinar. Have a great day.
Bye.